All right, let's get going. We'll stay on schedule. Pleased to help kick off this morning's session. We have a good lineup. I'm going to start with Young-Soo Lee from Waterloo, Canada. I've asked everyone to tell me, some secret skill or hobby. All I could get out of him was making a lot of kimchi. So that is enough for me to go visit. He's going to tell us about some anaerobic methane oxidation.
First of all, I'd like to thanks to Dr. Torres, the chair of this ISMET. I'm very honored to have opportunity to give a talk about a little bit new concept to you. Also, thanks to all committee member, Bruce Rogan, and Daniel, and I forgot other people's name. So. Today I will talk about a little bit a new thing, anaerobic methane oxidation coupled with extracellular electron transfer to conductive solid. Let me start with the issue of methane that we face. So I quickly searched for how many methane actually produce and consume on the Earth. That was 3.5 trillion cubic meter of natural gas in 2014. Now, I don't know how much that would be, but I feel trillion cubic meter, kind of wow. And it's really useful energy source for us if we can control it well. But if we cannot control methane production, like natural environment in former frost zone, then we know methane is strong greenhouse gas. Then we could have big problem later. The other issue is, as you know, we really want to improve sustainability in waste and wastewater treatment these days. Basically, we use methanogenesis-based wastewater, fermentation plus methanogenesis. So we like to claim we can treat waste and wastewater with small energy input, plus we can recover methane energy and reuse it for our society, then it can be sustainable. But what we found in this treatment system is accumulation of dissolved methane is not small, actually, it's big. As concentration 25 to 35 milligram per liter methane in the liquid when we treat dilute wastewater. BOD range about 200 to 400 milligram per liter. So if we want to accelerate this new concept of wastewater treatment, we need to control dissolved methane issue. Then we can deploy that technology. So we need to control undesirable methane, I like to call. First, methane production in nature, such as permafrost. Well, it's difficult to control, but at least we need to monitor methane production in nature. Then we can expect or estimate what's going on later. At least annual methane production in nature. Again, we need to control dissolved methane in sustainable engineered system. Then we can deploy this system in our society, then improving sustainability in waste and wastewater treatment. So that's why I start to have interest on AOM, anaerobic oxidation of methane. After I learned some about this biochemical reaction, AOM microorganisms more abundant in nature than I expect. So many papers, about 3 to 5, to my knowledge, they reported about 5 to 20%, recently 27% of total methane production in nature consumed by AOM. So that means it's not small, actually. So AOM could be dominant. Then based upon this fact, then I start to imagine, then we could engineer AOM for us. So I'm interested in MET always. So I start to imagine, can I engineer AOM with MET? So first goal is methanogenesis happen in form of prostron or remote area, for example. Then if we can monitor methanogenesis reaction, basically methane production, then give signal, but electrical signal using MET, that means we can monitor methane production in remote area. We don't need to visit there. So that is one goal, in situ monitoring system for methanogenesis. Second is, I really like to control aqueous methane in anaerobic membrane bioreactor or anaerobic wastewater treatment. That's motivation. So then I put my one big hypothesis. Normally AON coupled to sulfate or nitrite. These people found nitrate too, but not single. Actually, they work together, many. So I put one hypothesis. We probably have AOM microorganism that can oxidize methane to carbon dioxide. And we don't know this guy, but I just imagine. Then electron directly transferred to anode respiring bacteria, grow on the surface of electrode, or it can cause direct electron transfer between two microorganisms. That is one possibility. The other possibility, probably AOM microorganism generate C1 or C2 intermediate compound from this anaerobic methane oxidation, then ARB utilize it. That's second possibility for me. Then I and my student start to enrich geobacter culture on biofilm. That's easy for me because I just used to that. Using assay medium, it's kind of normal routine in MET lab. So we confirm geobacter is more than 70% using power sequencing. And then I didn't put anaerobic digestion sludge. I put RAS based on my experience because I need some unique special methanogens that can do AOM, not normal methane production. That's why I put RAS. Then we operated this system only with methane gas as electron donor. Then we realized that very small oxygen diffusion through tubing can create micro-aerobic system. Then, okay, we decide, all right, we need to move all system inside of anaerobic chamber to stop any oxygen issue. So we start to operate all system inside of anaerobic chamber. Then fix anode potential at minus 0.4 volt versus silver chloride. And again, methane 99% gas as used as sole electron donor and mainly carbon source, but we also put bicarbonate about 5 millimole. Basically, because of pH issue, I want to maintain neutral pH during reaction. Although this reaction is super slow, really, really slow. So we did a lot of tests for 4 years. So I compressed 4 years outcome in 20 slides today. So I will skip isotope test and NMR analysis, but we're focused on 3 things: stable isotope proving and combined with DGG with C12 and C13 methane. The second is fish targeting AOM archaea. We confirm they are archaea, not bacteria for AOM. And then ARB, actually geobacter. Then finally, we want to understand a little bit what would be main mechanism for AOM coupled to EET. And again, this reaction super slow, really, really slow. So typical chemical analysis didn't work for us. So we need something new. Then I decided to use metagenomic then. So we can identify some functional gene responsible for AOM or EET. We'll skip that. So basically, basically, or briefly, about 50 million sequence, that is starting point. Then about 30 sequence used for taxonomic classification. There are very complicated processes for screening sequence, which I don't know much about. So my collaborator in Yonsei University did this job for me. Then other part actually used for functional annotation related to AOM or ET. So first, current. That is direct evidence and easily get for our lab. So we operated this system over 750 days. Very consistent. You can see this peak actually a capacitance effect. So when we replace membrane or when we replace medium or something small inside of MET, then we need to disconnect it. Then once we connect it, then we always found like huge current, and that's capacity effect, we know that. So that's why you have like regular peak current because of capacitance effect. But basically we have very, very stable sustainable current, but very small too. So this is about 0.01 amps per square meter. And endogenous decay current for this without any methane is about 0.002 to 0.003. We know that number. So from this current profile for 750 days, I'm sure, okay, we have ET reaction from methane, but anaerobic condition. So we want to confirm this is originally from methane oxidation or not. So we did sparging test, kind of switching methane to nitrogen and nitrogen to methane. So now highlighted, orange highlighted means nitrogen sparging. So you can see the current is now milliamps per square meter, drops significantly. Then when we start to feed methane gas, then current go up again. Then nitrogen sparging go down. So we have reproducible a profile about this pattern over 10 times. Then we very sure AOM occur coupled to EET in our system. So we have AOM bug and ARB. Then we want to know who actually could do that job. So we compare SIP with combined with DGG. So basically C12, C13. We don't know why there are different profiles between C12 and C13. We need to study more. Right now, we don't understand. But we found a little bit different profile. But the common thing is green one indicate Geobacter. And then the red one, the dotted red one, Methanobacterium, here. Also, Methanocopopuscum, which was this one, too. So from this SIP test, combined with DGGE, we realized Geobacter would be main player, and methanogens, probably Methanobacterium, would be important player for our system. Together with that, we also learned one very important thing. The presence of C13 in DNA in Methanobacterium and Geobacter support exchange of intermediate carbon between AOM bug, we expect Methanobacterium, and ARB, which is Geobacter. If direct electron transfer happen, then we may not see any carbon in Geobacter, for example. See, when we label methane with 13. But we always found big band, thick band for Geobacter in SIP DGG test, which means we don't know which compound, but AOM bug do something, then produce some small compound for ARB, then ARB happy to utilize it for current generation. That's my perspective and interpretation of this test. So I don't want to say direct electron transfer between Methanobacterium and Geobacter never happened. I don't want to say that, but our result support direct electron transfer can happen, but intermediate carbon actually happen to our system. Then we target, we want to visualize what happened in biofilm system in MET. So based upon SIP test, also pilot sequencing, I didn't show you. So pilot sequencing also show Methanobacterium for archaea and Geobacter for bacteria actually are dominant in biofilm system. So based upon two outcome, we design proof for Geobacter and the Methanobacterium. Now Geobacter is red, and then Methanobacterium is green. So you probably see that image or not. Maybe 3D image is much better. So you can see the center is electrode. And we have a red color things and the green color things and kind of live together. And the other image, that image would be better. So again, red represents Geobacter, green is Methanobacterium. So we conform two microorganisms live together, attached on the biofilm anode. So fish exactly support that. So third message is center of interaction between Geobacter and Methanobacterium exists in our system. And they probably grow together in biofilm anode. And that means they need some niche or reason for that. Now, after this confirmation and data interpretation, and I start to wonder then what will be AOM mechanism. We have two AOM mechanisms up to now. One is intracellular oxygenic AOM pathway. The other is reverse methanogenesis pathway. Then again, this reaction is super slow. So typical chemical analysis like direct measurement of O2 never tell us oxygen produced or not. So that's why I contact person about metagenomic. And they did a great job for me. So using C13 methane. When I use C13 methane as feed, and we grow them for 6 months, take biofilm sample, then they start to analyze this. So you can see now archaea Methanobacterium is dominant based upon Silva SSU RNA targeting. Then Geobacter is dominant for bacteria. So very consistent to pilot sequencing data and then SIP DGG outcome. And functional gene for AOM, and this is a pathway for reverse methanogenesis. So methane now basically just go backward to typical methanogenesis from CO2 to methane. So most important thermodynamic bottleneck, actually these two steps, catalyzed by MCR gene, MTR gene, almost 60 to 70 kilojoule per methane is required to drive this reverse pathway. So thermodynamically unfavorable reaction. That means AOM bug needs some external energy source. And we don't know what external energy source would be right now, but we found for archaea this abundant gene expression for archaea instead of bacteria. And then taxonomic identification for AOM, we found Methanobacterium from MCR to MTR gene, which is from here to here for Methanobacterium. And the other gene, we're still working on it to understand the implication of this huge data, but we found NHRACD for Geobacter and other bacteria. I'm interested in this ACD gene responsible for acetate production in reverse methanogenesis pathway. So we don't understand what's the relationship between this reverse methanogenesis and AC gene in very detail, but somehow we found Geobacter is responsible for that. The other functional gene related to ET, then we found C-type cytochrome, outer membrane protein, nanowire production, mainly for Geobacter. So this metagenomic analysis strongly support Geobacter is truly ARB for my system. So first message is, yes, based upon metagenomics, reverse methanogenesis truly occur in our system. That's why methane oxidized to carbon dioxide anaerobically. Then we identify key enzyme for reverse methanogenesis from MCR gene to FMD. Then we also confirm methanobacterian species is main player for reverse methanogenesis. And then another important message is Geobacter is true ET player in our system. We also found key enzyme for ET, outer membrane protein, C-type cytochrome, and nanowire synthesis. So now, my conclusion from this study is AOM was coupled to ET for current generation in MET. Then syntrophy interaction between methanobacterium and Geobacter was built for AOM in biofilm. And stable isotope DGG indicate the exchange of intermediate compound between two microorganisms. We don't know direct electron transfer happen or not, but our outcome support they actually share some intermediate compound between two microorganisms. And we confirm reverse methanogenesis happen for AOM in our system. And we think intermediate generate from RM. We don't know which type of intermediate compound, but that is electron source for Geobacter to generate current. So now future study or my question, my personal question that I really want to answer. What kind of compound for intermediate, exchange intermediate compound? Now I start to suspect fomate based upon reverse methanogenesis pathway or acetate or others. Actually, NMR, thanks. NMR outcome support ethanol production. We have ethanol peak in NMR. And we don't understand why we have peak for ethanol in this entropy. Second question is, what is oxygen source? Okay, it happened, but what is oxygen source? We need oxygen source for methane oxidation to carbon dioxide. And we don't understand what is oxygen source right now. And can we engineer this new concept? And I'm a little positive, at least monitoring methane production in nature. And that's my old talk. And thanks for this student, Mr. Yahuan. Actually spent 4 years just for this and other funding agency. And thank you very much.
As I learn this, I get the first question. Just some really quick details about the way it was grown. In your anaerobic hydrogen, separated cell, electrode cell, or is it separated H-like cell? Hydrogen.
Okay, thanks. Yes, is dual chamber. We have separator between anode and cathode, yes. We have hydrogen in anaerobic chamber about, I forgot the exact percentage, but probably 5 to 10%, I guess. And we never check hydrogen actually diffused to our system or not. So very good point. I check.
Question: How labeled? So, you can make the argument if you have trace contaminant side, you have just methanogenesis going on. No reverse.
Can you?
So when I say the body composition I'm wondering what the purity is and what purity of the C13 methane. Yeah. If you have 0.01% contamination, it becomes C13 carbon monoxide. You can explain this by classical genesis and without any reverse thermogenesis. Because it's very sensitive, it's very slow. And actually, it comes from water.
Yeah. I agree. Totally agree. We never thought about that possibility for our system. I think we need to check after I come back to my lab. But the current profile and other outcome that I didn't show you actually strongly support reverse methanogenesis, actually. We also use this inoculum for sulfate and nitrate, and they work, actually. So we are very positive about reverse methanogenesis. But yeah, that's very good question and comment. I think I need to conform carbon monoxide. Yes.
So I've got two small questions. I was also thinking that you're making hydrogen at the cathode and it's slowly diffusing across, and then you have normal methanogenesis. Do you actually have a clear relationship between the methane consumption and the current? I mean, that's the key.
That's one.
We
Okay.
tried for years to do methane oxidation. Daniel too, clearly.
Every
Many-
time I tried it, I found there was carbon monoxide in some
Yeah.
of my stuff. in my
So that causal relationship will be essential.
Mm-hmm.
Two, what did you flush with? Nitrogen gas?
Flush rate.
Yeah, just with nitrogen gas.
when you,
Yeah.
when you did your
Because if you flush just with nitrogen gas, you're flushing out all your bicarbonate and you'll shock the cells. The anaerobic cells need carbonate around them.
Okay.
And so all microbial activity under anaerobic conditions just falls flat if you flush with pure nitrogen.
All right.
It needs to be nitrogen CO2.
Right. So about first question, again, this is 750 days outcome. We try so many type of MET. So we have like this long H type, same outcome. And there is no direct relationship between diesel methane concentration and current. Main reason is AOM rate is super slow. So even though diesel methane concentration super high, their rate is just flat. So that means if we have a ARB that can directly oxidize methane and generate current, we would have direct relationship probably. But if we have syntropy inaction and then first step is rate limiting step, then we cannot get it. Question number two, we try nitrogen and carbon dioxide too. So 80, 20%,
Yeah.
same outcome.
Yeah, I think we could do this all day. I'm on a 2% schedule, so really exciting stuff. Thanks, Peter.
Thank you very much.